having an exam in science.
What? Exam?
Yes.
Oh my god.
I didn't review anything.
So let's start reviewing now.
Okay, before...
Kung wala ka nang maintindihan, kung wala ka nang makapitan, kapit ka sa akin, kumapit ka sa akin.
Our lesson for today is about central dogma. Do you know what is central dogma? You. Okay, now open your books on page 169.
Cells, like these prokaryotic replicate themselves quickly and efficiently, part of the process of asexual reproduction and the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the accurate process of DNA replication to ensure future generations of cells will have the same genetic instruction as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of the four nitrogen bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine and thymine forms 2 hydrogen bonds with adenine. This is referred to as a complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing is the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with a double-stranded DNA being separated and each origin strand called the parent strand is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides in the 3' end of the newly forming strand. DNA replication will begin at a specific area at the molecule called the origin of replication. Denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first study replication in prokaryotic models like E. coli. A number of enzymes are needed for proceed once the replication fork is established. Helicases separate the strand of the double helix and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand Other proteins such as beta clumps and clump loader help hold the DNA polymerase in the place of DNA. Short sequence of RNA called primers have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotide without a primer. Replication of both strands occurs at the same time. One thing continues synthesized and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end moving continuously towards the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in the segments called Okazaki fragments. Replication on these strand uses primase to add polymerase 3, then adds short sequence of the nucleotides, the Osaki fragments, to the primer filing in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase 1 replicates the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once the both leading and lagging strands are completed, the replication of two identical copies of DNA molecule result. This process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Bacterial population can grow and increase the number of individuals in a colony.
In order for our bodies to function, we need to supply them with the varieties of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used or as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of a cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of an enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then, during the elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecules. Once the RNA polymerase reaches the terminator portions of the gene, the messenger RNA transcript is complete and the RNA polymerase, the DNA strand and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns in order for the messenger RNA to be used in translation. The non-coding introns need to be removed and modifications such as a 5' cap and 3' poly A tail are added. This process called intron splicing and is performed by a complex made up of proteins and the RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is a transformation in the messenger RNA strand translated into a protein?
Nitrogenous bases are grouped into three-letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions, called the E, P, and A sites. During elongation, individual amino acids are brought to the mRNA strand by a transfer RNA molecule through complementary base pairing of codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charge transfer RNA molecule binds to the A site and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecules at the P sites. The complex slides down one codon to the right where the now uncharged transfer RNA molecule exists from the E sites, and the A site is often to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site and a stop codon and the polypeptide is released from the transfer RNA reach. A release factor binds to the A site at a stop codon and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can resemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modification takes place in different organelles for different proteins. In order for a digested enzyme to be secreted into the stomach or intestine, the polypeptide is translated into endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell, the lumen of the digestive tract. Proteins are needed for most physiological function of the body to occur properly, such as breaking down food particles in digestion, and the process of transcription and translation make the production of proteins possible.